Hello everyone. In this video I'd like to talk about the regulation of gene expression, or what's more commonly referred to as gene silencing. Now, you can have transcriptional, post-transcriptional, translational or post-translational gene expression regulation. So first of all, what we're going to do is we're going to put in a sequence of events, if you like. We're going to start with DNA. Here's some DNA in the cell. Now, after transcription, and I talk about transcription and translation in a separate video, we end up with some messenger RNA. And then after process of translation, we end up with a protein that we want. Now what I'm saying is that we can regulate at any stage of this process. So if we regulate here, this is called transcriptional regulation. So this is transcriptional regulation. If we were to regulate at this point here, that would be post, and I'm just going to put TR for transcriptional regulation just to be a bit quicker. So that would be post transcriptional regulation. If we were to regulate at this point, this would be translational regulation. So you might be able to guess that if we were to regulate at this point here, this would be post translational regulation. So either before transcription and translation, or after, essentially. So that's what we're going to do. We're going to look at those in turn. Let's think about, first of all, transcriptional regulation. So I want to talk about this one first. So what I might do is, just to keep it all on the one screen, we'll move this over here. So we're going to focus on this first, transcriptional regulation. So let's have a simple transcription model first of all. So let's imagine here's a piece of DNA. There's a block of DNA. I've got a particular gene here. Now we have what's called RNA polymerase, which is going to, so RNA polymerase is going to help initiate transcription. Now what happens is that RNA polymerase would bind to the site, work its way down, and we would get transcription of that strand, and we get a piece of messenger RNA produced. Now what we have are what are called transcription factors. So this is key. There is something called transcription factors. Now, transcription factors are molecules that move from the cytoplasm into the nucleus to initiate transcription. So when we have transcription, we're obviously referring to the genetic material. So we have our transcription factors move from cytoplasm into the nucleus to help initiate transcription. Now, each one has a specific binding region, and when it binds, transcription is stimulated. So if we wanted to be more specific on this picture, what we could say is that there's a particular region here in red. Now that red region there would be our binding site. And we have, and I'll colour this in green just to make it a bit different, we have in green transcription factor. In fact, and I'll put a little green box over here just to remind you. So when that transcription factor comes and binds to the binding site, then RNA polymerase can work its way down that piece of DNA. So that's what transcription factors do. And in fact, we can get something called a promoter transcription factor complex, and that just helps to ensure that we have an extra sort of level of regulation, if you like, of transcription. So I said that we can have a transcription factor binding to this binding region. There is also another region called the enhancer, which another transcription factor can bind to, and that just, as the name suggests, enhances transcription. But what we're going to do is look at how we switch genes off, because what I've just drawn for you is how to switch a gene on. If a transcription factor binds to a binding site, then RNA polymerase works its way down, we make mRNA, we make a protein, that gene is operating, it's on. Because we're fulfilling its function, if you like. We're synthesizing that polypeptide that's going to be turning into a protein. Let's think about when we turn genes off. Now, when we turn genes off, what we have is something called an inhibitor molecule. And an inhibitor molecule, so I've coloured it in blue here, an inhibitor molecule would bind to that transcription factor. So this is our inhibitor molecule, and that could bind to the transcription factor. Now, if that happens, clearly RNA polymerase can then not bind, so we don't have any transcription. So this would be a transcriptional method of regulation. We turn the gene on by having a transcription factor bind, but if we have an inhibitor molecule actually bind to that transcription factor, then it's not able to connect with its binding site. RNA polymerase therefore isn't initiated, so we don't get transcription, we don't get that mRNA forming. There is also something called, as well as inhibitor molecules, molecules called repressor molecules. Repressor molecules can actually bind to the binding site itself and also stop transcription. So that's a way to turn genes off. You have an inhibitor molecule bind to the transcription factor, or, and I'll just put this down at the bottom because it's an important thing to know, you can have what are called repressor molecules actually block the binding site itself, and that too can turn genes off. Let's think about how we can turn them on again, because clearly once we've had an inhibitor molecule bind to a transcription factor, to turn it on we're going to need to get rid of that inhibition. Now, molecules such as estrogen, molecules like estrogen can diffuse through lipid cell membranes. Because they are lipid soluble, they can diffuse very easily through a lipid cell membrane and into the cytoplasm. And there, from the cytoplasm, can come and bind to receptors on transcription factors. And when the estrogen binds to the transcription factor, it changes the shape of that transcription factor. It loses the inhibitor molecule falls off, and the transcription factor can once again bind. So let's just put that into place. So what we've done is turn gene on first of all, and then I explain how you turn it off with an inhibitor molecule. So now we're going to move, take the story up here. And what we're saying is, if we imagine here, just draw again, here's our original piece of DNA, here's our RNA polymerase enzyme, here's our binding site, which we need our transcription factor to bind to. So we have a transcription factor here, but we had our transcription factor inhibited by inhibitor molecule. Then you can have estrogen, or molecules like estrogen, bind to the transcription factor, and when it binds to the transcription factor, it changes its shape. That causes the inhibitor molecule to fall off, and the transcription factor can now go and bind to the binding site, and that will cause RNA polymerase to clearly work its way down the DNA and lead to transcription. So I've talked about in this video how you turn the gene on, turn it off, and then even turn it back on again. What I want to finish this video with is by looking at post-transcriptional regulation of gene expression. So I want to look at now this point. I could go into detail with all four, but I want to particularly pick up on these two. So what I've talked about so far is transcriptional regulation, and I want to look at post-transcriptional regulation of gene expression. So I'm going to do this on a separate slide. Okay, so in post-transcriptional regulation of gene expression, we're going to be dealing with what's called RNA and then a little I. Now that stands for RNA interference. Because what we have is ultimately the interference of RNA. So we're talking about RNAi interference to regulate gene expression once transcription is complete. Now what we have in the nucleus of a cell is a piece of double-stranded, and I'm drawing this very rough, double-stranded mRNA, messenger RNA. Now when you're taught about this, you're normally taught mRNA is a single strand of nucleotides, and that is true. But there is a special molecule of double-stranded mRNA. So let's label that. This is double-stranded mRNA. And what we actually have is an enzyme called the dicer. So an enzyme called dicer does as kind of the name suggests, dices this up. It works its way along this messenger RNA and breaks it up into smaller pieces that look a bit like this. And these are called short interfering RNA segments. So SI for short interfering RNA. So just put there, so this is short interfering RNA. So now we've made small pieces of short interfering RNA. We're going to use another big enzyme, and this one is called RISC. And that stands for RNA-induced silencing complex enzyme. So I might just very quickly just write that down so you've got the details. So it's RNA-induced silencing complex enzyme. Some specifications require you to know what RISC stands for, some don't, but that's what it stands for. Now what happens ultimately is that short interfering RNA, one of those small pieces that we had, comes to join to RISC. it binds to risk, and then what we have is half of that double-stranded mRNA detach. So ultimately, we have, if we were to draw it a bit further on, here's our enzyme. We have double-stranded short interfering RNA conjoin, and then half the strand moves away. So we're left with only a half mRNA strand, looking a bit like this. Now, if we take our messenger RNA, here's our messenger RNA that we've already transcribed, because remember, this is post-transcription regulation. Here's our messenger RNA. What we find is that that short interfering RNA guides the risk enzyme to the messenger RNA by lining up complementary base pairs. So, and we'll just cover this in purple, because of complementary base pairing, this short interfering RNA strand on the risk enzyme can find its complementary pair on the messenger RNA. That risk enzyme, when it moves down to the messenger RNA, what it actually does, once it's there, is cut the messenger RNA into smaller pieces. So that Si, short interfering RNA, guides the enzyme to the messenger RNA by lining up complementary base pairs, and every time that those pairs meet, the risk enzyme cuts the messenger RNA. It's like taking a pair of scissors to it, cuts it into little bits. Now if you do that, you are no longer able to translate that messenger RNA into a polypeptide. So we have therefore silenced our gene. The gene is not expressed because we have stopped that messenger RNA becoming, ultimately, it's protein. So we've been able to regulate gene expression and silence the gene by cutting our messenger RNA. Now, two questions that students often ask is, what could be the benefits of doing this? Two really important benefits, and that's what we're going to finish on. So I want to just very quickly, at the top, I'm just going to put them here. The benefits, because the benefits are really important to consider. The benefits of using Dyson in this particular method. The two key benefits. One of them is to identify the role of genes by observing the effect of blocking them. So you can identify the role of genes. If you block a particular gene, if you stop that messenger RNA coding for a particular protein, and you kind of watch what happens, you can see what that gene does. The second benefit is that we can actually block genes using short interfering RNA to prevent certain genetic diseases. So we can actually block genes to prevent disease. And a good example of that, one that many examples like to comment on, is Huntington's disease. Now, DNA that codes for short interfering RNA can be introduced, and the short interfering RNA is produced that is specific to messenger RNA, mRNA, made by the Huntington gene. So when the Si, or the short interfering RNA, joins with the RISC protein to form a silencing complex, that complex can join to the Huntington mRNA and destroy it. So it means that the Huntington mRNA cannot be translated into the Huntington protein, and that would help to reduce the symptoms of the disease. So that's just two of the benefits of this post-transcriptional gene expression regulation. Okay, I hope all that helps.